Hello everyone, and welcome to another recorded lecture of BIO101 online. Today we will learn about Chapter 9, which is the cell cycle and cancer. So the cell cycle describes the process of cell growth and cell division. And what you'll see in cell division is that you have one parent cell at first. So we could say this is the parent cell. And this divides into two daughter cells. So this is actually showing the first cell of an embryo. And during embryonic development, what starts as one single cell becomes a multicellular organism. So the cell divides in a very controlled process called the cell cycle. For each of the daughter cells from the division to have identical DNA, the parent cell must first replicate its genome. Right, so if you want the daughter cells to have the identical set of genes, you have to first make a copy of the parental DNA, so then the daughter cells can each have an identical copy of the genome. I'm going to talk about prokaryotes first and kind of get them out of the way, because their cell cycle is very simple. Prokaryotes divide by what's called binary fission. And since prokaryotes are only one cell, when they do cell division, they're doing so to reproduce. Right? Every time that they divide, they're reproducing. This is a form of asexual reproduction. In binary fission, the prokaryote will duplicate its DNA and then split into two identical cells. So if you recall, prokaryotes have a single circular chromosome. And what happens is this chromosome will be duplicated. So we have double the amount of genetic material. The cytoplasm will also expand. And what you'll see is that the two halves of the genome, right, the two copies, get allocated to separate sides of the cell. And as the cell elongates, there's a little cleavage over here, which will divide the cells into two. So this cell is identical to this cell, which is identical to this cell. Right? So this was the parent cell, or the parental cell, that divided into two daughter cells. That's binary fission in prokaryotes. Let's move on from there. In eukaryotes, the cell cycle is a continuous process with many distinct phases. So the cell cycle is like a clock, and each of your body cells is in one of these phases right now. So we're going to talk today about the cell cycle and what each of these phases do and why they're important. Before we talk about that, I want to break down the two types of eukaryotic cells we have in our bodies. So in multicellular organisms, there are two types of cells. Most of your cells are somatic cells, which means body cells. And body cells contain two sets of chromosomes. Because they have two sets of chromosomes, we call those cells diploid. You could think about di for two. So somatic cells, body cells, contain two sets of chromosomes. These cells include your hair cells, your eye cells, your skin cells, muscle cells, blood cells. All these cells contain two sets of chromosomes. One set came from mom, the other set came from dad. So those are somatic cells or body cells. So every cell in your body is a somatic cell except for gametes. Gametes are sex cells like sperm or egg, and those only have one set of chromosomes. We call those haploid. 1N. And what that N means is the number of chromosomes we have. So for humans, N is 23. We have 23 pairs of chromosomes. So in your hair cells and your skin cells, you have 2N, or 46 chromosomes. 23 came from mom, 23 came from dad. In sperm or egg cells, you only have 23 chromosomes. And we'll talk about how those gametes get made in the next chapter. So just as an overview, right, body cells have two copies of every gene. We call those somatic cells. Gamete cells have one copy of every gene, and we call those haploid. as opposed to diploid. So what we'll talk about today is how somatic cells divide. So somatic cells, our body cells, divide by mitosis and cytokinesis. They do this for growth, repair, and development. So as part of regular embryonic development, cells need to constantly divide. So as a baby becomes an adult, there's a lot of mitosis that has to happen. Also, if you trip and scrape your knee and you damage some skin cells, your skin cells have to do mitosis to replace the ones that were damaged. There are two parts of the cell cycle, the somatic cell cycle. There's mitosis and cytokinesis. And mitosis refers to division of the nucleus that contains DNA. Cytokinesis refers to division of the cytoplasm. So together with mitosis and cytokinesis, we're going to see how somatic cells divide. Interestingly, when I say that cells are dividing, that means they're also multiplying. So biology is the only place you're going to see where multiplying and division are the same thing. Right? Cell multiplying is cell division. The key point about mitosis is that it generates genetically identical cells. So if you start off with one cell, you're going to end up with two cells that are identical copies of the original. And remember, only somatic cells multiply by mitosis. So gametes, like sperm or egg, multiply by meiosis or meiosis. So once we have adults at puberty, they could generate gametes, like sperm or egg, by meiosis. Once sperm and egg fertilize each other, that fertilized egg can undergo mitosis again to help develop the embryo. So all of our somatic cells have exactly the same genes. They're identical to each other for the most part. That's important to keep in mind. And most of your cells are not dividing right now. And somatic cells that are not dividing in mitosis or cytokinesis are in interphase of the cell cycle. So I'll talk a lot more about interphase in a bit. So mitotic cell division occurs in somatic cells and results in genetically identical daughter cells. A cell's endowment of genetic information, right, in the form of DNA, is called its genome. And like I mentioned before, cells have to first duplicate their genome before they divide to make sure that each daughter cell will receive an exact copy of all the genetic information needed to survive. In addition to DNA, cells must also duplicate their organelles and all the cellular enzymes before they can divide into two. So what we will see over the course of this lecture is that when the cellular environment is just right, the two copies of DNA will be allocated to separate ends of the cell. They're going to be, we call those poles, like the North Pole and the South Pole. There's two different poles to the cell. So the DNA copies will be separated to opposite poles of the cell, and then a signal will be sent to split the cell into two daughter cells. That's the process we're going to see today. So what started off as a parent cell, in interphase, right? The enzyme DNA polymerase will copy all the genetic information before mitosis begins. So we start off with a parent cell. DNA polymerase is the enzyme that copies all of the genetic information in this parent cell. So here we have double the genetic information. And then during mitosis, we're going to see that this splits into two daughter cells. So we said there's interphase, mitosis, and then cytokinesis. So the genome is normally found in the form of chromatin. So chromatin is a fancy way of saying DNA wrapped around proteins. So we know inside of our nucleus, we have our genome. The genome consists of DNA, like the double helix, but it's wrapped around certain proteins. and I'll talk about the importance of these proteins in a bit. But normally, in a non-dividing cell, chromatin is very loosely packed. So when genes are active and the cell is not dividing, the DNA is loosely condensed, and we call this euchromatin. So when it's loose yarn, like loosely packed DNA, we call this euchromatin. But after DNA gets replicated, we have to condense this chromatin. This is really hard to divide into two, loose yarn, right? So what happens is we're going to see this euchromatin gets condensed into heterochromatin to form visible chromosomes. So what you notice in a picture as chromosomes is really very, very, very highly condensed DNA. Most of the time, your DNA looks like this. Only during or right before mitosis does the DNA condense and look like chromosomes like that. So euchromatin is loosely condensed. Heterochromatin is very tightly condensed. So we start off with DNA, which is made up of nucleotides. Right? Nucleotides are sugar, phosphate, and a nitrogenous base. So this DNA double helix gets wrapped around these proteins called histones. So as DNA wraps around histones, it gets more tighter and more tightly and tightly compact. So this gets wrapped around itself, wrapped around even more and more and more, until we have this really highly compacted chromatin. And this is what a replicated chromosome looks like. So normally, we only have one of these chromosomes, but it gets replicated during interphase. We call each of the halves of this replicated chromosome a sister chromatid. So these two halves of the replicated chromosome are sister chromatids. So this sister chromatid is identical to this sister chromatid. So let's go over the parts of a chromosome, right? So remember that chromosomes only appear this way during the first phase of mitosis, after replication. Most of the time, your DNA is uncoiled like loose yarn. But before mitosis can begin, or I should say during mitosis, we're going to see that the chromatin gets wrapped into chromosomes. And when it's replicated, we see that there are two halves of the replicated chromosome. Each is called a sister chromatid. So this is one chromatid, here's another chromatid. At the end of each chromatid is called the telomere. So these are telomeres at the tip. So you can remember T for tip, telomere. Telo always means the end. The centromere is the region in the middle. So centromere is in the center, telomeres are at the tips. And there are special proteins that are linked at the centromere called kinetochore proteins. Kinetochore proteins. So these kinetochore proteins will be very, very important in mitosis because we're going to see that microtubules can grab onto these and pull chromosomes to opposite poles of the cell. So once more, I'm going to review, right? We start off with what is one chromosome, but in reality, if this were our cells, we would have 46 of these, right? Remember, we have 23 chromosomes from mom, 23 chromosomes from dad. So we have 46 of these. During interphase, each of the 46 will be duplicated. So each of the 46 chromosomes will then have a sister, which is an identical copy that's held together at the centromere. During mitosis, what we will see is that the two sister chromatids get pulled apart. So we have two identical copies of genetic information. So once we separate the chromatids, how do they travel to separate ends of the cell? Right? Microtubules, remember that's part of the cytoskeleton. So microtubules form what's called the mitotic spindle. And the mitotic spindle controls the chromosome's movements during mitosis. So the mitotic spindle is very, very important. That's shown in pink here, these microtubules. They can pull on the chromosomes so they can separate to opposite ends of the cell. So from where in the cell do microtubules come from? Do you remember? So the centrosome. The centrosome, right, is the microtubule organizing center in animal cells. And the centrosome contains centrioles, and that's where microtubules come out of. So during replication, during cell division, we're going to see the two centrosomes, the centrosome duplicates itself, and they're going to go to opposite sides of the cell. So normally we have one centrosome, but before division, we need to make a copy. So we have one centrosome that will be on one side, another centrosome that will be on the other side. So here's a list of some confusing terms that you can pause and look over. I think flashcards are great just to make sure that you don't get confused between any of these vocab terms. So where would centrioles be found in an animal cell? So you could pause here. And the answer is C, the centrosome. So this is a picture I actually took when I was working in a lab a few years ago. And this is immunofluorescence. So what I was able to do was take cancer cells and use specially labeled proteins called antibodies to detect other structures in the cell. And they would light up different colors. So I would like for you to pause and try to guess, what did I stain in blue and what did I stain in green? I'm not going to tell you the answer until later, but try to figure out, these are cancer cells. What would be in blue and what would be stained in green? So now we're going to dive into the eukaryotic cell cycle. So the cell cycle consists of three parts. Interphase, that's where the cell is going to grow, copy its DNA, and just prep for division. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. So as you're watching this lecture, make sure you're taking notes. And I would organize your notes as an outline. So we know there are three phases of the cell cycle, or three parts of the cell cycle. There's interphase, mitosis, cytokinesis. Each of those will have subsections. So again, make sure you have like a neat outline that shows how all these phases fit into one another. So interphase. Your cells are in interphase 90% of the time. So most of your cells' lives are spent in interphase. An interphase has three parts. It starts with G1, which stands for the first gap. Then there's S phase, which stands for synthesis. And this is where DNA polymerase will synthesize new DNA. It will copy the genome in S phase of interphase. And finally, there's a second gap phase called G2. So interphase has G1, S, and G2. And cells spend most of their time in these three phases. So starting here, interphase, G1. This is where the cell is going to do most of its growth. It's kind of hanging out, waiting for a signal. Then an S phase over here is when DNA gets synthesized by DNA polymerase. The cell then enters G2, where the cell continues to grow, enzymes are produced, and then you can enter mitosis and cytokinesis. So this whole thing is interphase. And we'll talk a lot more about interphase soon as well. So let's watch this quick video on interphase.
The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap 1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis.
So now we will dive into mitosis. So mitosis is divided into the following phases. There's prophase, a short step called prometaphase, metaphase, anaphase, and telophase. So mitosis, when I learned it, was always P.M.A.T, prophase, metaphase, anaphase, telophase. Since I've learned it, there's a middle phase put in between prophase and metaphase. So it's called prometaphase. I'm not going to ask you specific questions too much about prometaphase on a quiz, because it's kind of a mixture of the two. And I'll talk about what you have to know, right? So what's on the slides are what you have to know more so than what's in the book. But you must do the reading in order to understand what's on the slides, because I do a lot of summaries. So, cytokinesis, which is division of the cytoplasm, starts during late telophase, and that's when we see two new daughter cells form. Okay, so let's start with prophase of mitosis. So, before prophase, the cell looks like this. The nucleus has DNA that's unraveled. There's one centrosome with microtubules. But what we're going to see in prophase, sorry, there's two centrosomes, but they're together right now. So what we're going to see in prophase is that the centrosomes will start to move to opposite sides of the cell. We're going to see that chromatin condenses into chromosomes. So look over here in this animated picture, all the chromosomes are unraveled here. But what happens is that these the unraveled chromatin will condense into visible chromosomes during prophase. In prometaphase, we see the nuclear envelope break down, so there's no more nucleus, and the centrosomes begin making the microtubules for the mitotic spindle. So a lot happens. I would say actually, if you remember prophase, you have most of mitosis remembered. So in prophase, you have the chromosomes condense, right? We see like that. The centrosomes start moving apart. In prometaphase, the nuclear envelope will break down, and the microtubules will start to form the mitotic spindle. If you remember, this is a screenshot from the Inner Life of the Cell video. And this is the centrosome. You can see two centrioles, and all of these string things are the microtubules that are coming out of it. So next we have metaphase, where chromatid pairs meet in the middle. You can remember M, metaphase, meet in the middle of the cell. Specifically, microtubules attach to the kinetochore proteins at the centromere. So remember, we have two identical sister chromatids. They're all going to line up at the equator of the cell. So they line up right at the middle. It's really important that they meet in the middle so the cell can divide the genome equally. Right? We want equal division. So it has to meet exactly along the equator. Each sister chromatid will be pulled by the spindle fibers at the kinetochore. So this will move this way, this will be pulled that way, and the sister chromatids will separate. So you can pause here, and the answer is B. So anaphase. Anaphase is when the two sister chromatids of a duplicated chromosome are pulled apart. You can remember anaphase A for apart, because that's when the two sister chromatids are separated. Each chromatid will be moved toward one end of the cell. So you can see over here, all the blue chromosomes are being split, The microtubules are shown in green, and they're pulling the chromatids to each half of the cell. In telophase, which is the last phase of mitosis, we see is the reverse of prophase and prometaphase. So whatever happens in prophase and prometaphase, the opposite will happen in telophase. So we're going to see that the chromosomes decondense into chromatin. They no longer look like chromosomes. We're going to see the nuclear membrane will be rebuilt around the two sets of DNA. We'll see the spindle disappear, right? There's no more mitotic spindle anymore. So everything that happens in prophase is kind of undone in telophase. We rebuild the nucleus, right? We decondense our chromosomes, and we put away our mitotic spindle. At the end of telophase, there's a formation of what's called the cleavage furrow. So in animal cells, there's a shallow groove that cinches the middle of the cell, right where the chromosomes lined up. So the chromosomes in metaphase lined up here, and that dictates where the cell is going to divide. And of course, this happens in cytokinesis, right? So division of the cytoplasm occurs by cleavage in animal cells. And what allows for this cleavage is actually microfilaments, which are made of actin. And they form what's called a contractile ring. So it's almost like a purse string. Like when you pull on this little string, it'll cinch the membranes together so the two daughter cells can be separated from each other. So that's because of actin. Actin will pinch the cells, and then you'll have two identical daughter cells. Plant cells don't do this whole cleavage furrow with the contractile ring. They form a plate of cell wall right between their two cells. So here is an overview from the book of each phase. You should be very, very familiar with the animated pictures as well as the actual images from under a microscope. Right? These are really good, succinct descriptions of what happens at each phase. So definitely pause here and make sure you understand what's happening at each phase. Make sure you can put them in order. And make sure that you're able to make the connection between the actual image and what you're seeing in the cartoon. So here is a cell that's in mitosis that's been stained with certain dyes. And the chromosomes are stained in blue, and the microtubules are stained in red. So what phase does this look like? And what phase does this look like? Right, so this is metaphase, and this is anaphase. So here's a picture of real cells dividing.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
Great. So this is one of the best animations I could find on mitosis. I will include the link on Blackboard. So please make sure you watch this very, very carefully. I'm going to play it now, but you must watch this several times. And I would not go any further until you really understand what's being shown in this animation.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
So now we will talk about cell life and cell death. So, a cell's longevity, like how long it lives, and the frequency of cell division varies greatly. So this depends on the type of cell it is, how healthy the cell is, and what's going on around that cell. All right, so all these factors will determine whether or not a cell is ready to divide, or whether a cell should just hang out, or possibly whether the cell should die. The cell cycle is under very, very, very tight control to make sure that cells have the right amount of balance between life and death. So how does a cell know when to divide or not? Right? How does a cell know when it's appropriate to undergo mitosis and cytokinesis? Of course, cell signaling pathways help us. So we know that on the plasma membrane there are several receptor proteins that can perceive what's going on the outside world and transmit that information to the inside of the cell. So there are several signaling pathways that can tell the cell when to divide and when not to divide. One such signal are called growth factors. So growth factors are special signaling molecules. So this little triangle is an example of a growth factor. They bind to receptors on the plasma membrane and stimulate cell division. So when this growth factor binds to this receptor on a cell, the cell will know that it's time to divide. So that's how signals tell the cell when to divide or not. So let's take a zoom into the cell and really think about what's happening here. So inside the nucleus, of course, there are these genes that encode for mitosis, so to speak. I'm simplifying here. But there are genes that tell the cell to divide, but they're usually not turned on because we don't want the cell to always divide. However, when a growth factor binds to this receptor, something on the inside of the cell changes. So when the growth factor binds to the receptor, there's a change in the structure of this membrane protein that allows it to activate other proteins inside of the cell. So this growth factor activated all these other proteins inside of the cell, which will end up sending a special factor called the transcription factor inside of the nucleus. And this will tell this gene to be turned on. So because of the growth factor, this growth gene will be turned on, and then it will make proteins that will stimulate cell growth and mitosis. So this is important. You could go back and review, but normally this protein that stimulates cell growth and mitosis will not be made. It is only made when a growth factor can bind to a receptor, and that signaling pathway can tell the growth gene to be turned on. Once this protein is made, then the cell will undergo division. If the protein is not made, the cell will not divide. Inside of the cell, there are other proteins called cyclins. And cyclins are signaling proteins that trigger the various phases of the cell cycle. So cyclins are called cyclins because their concentrations increase and decrease. They cycle over the course of cell division. And there are different cyclins. So for example, I could just say that only when cyclin E is at its peak can S phase start. All right? And then only when cyclin B is at its peak can mitosis start. So these cyclins go up and down, and these dictate the concentration of these different proteins, dictate which part of mitosis should be triggered. I'm going to give you a little more information than you need to know, but these cyclins end up pairing with special kinases that activate phases of mitosis. We spoke about kinases as being like the on switch. Kinases can activate other proteins by adding a phosphate. So these kinases are only functional when they have a cyclin present. So these are called CDKs. They're cyclin-dependent kinases. They're dependent on cyclins. So these CDKs are always present in the cell, but only when a certain cyclin is present will this kinase be activated. And when this is activated, it can turn on other proteins in the cell that are required for a certain phase of mitosis. Again, not that easy. This is the bare surface of cyclins and cell cycle control. You could spend months talking about literally just cyclins and how they pair up with CDKs. So right now I want you to know that cyclins are proteins that fluctuate inside of the cell and they trigger the different phases. And they can pair up with CDKs, which are always constant, but you need to have a certain cyclin in the cell at a high enough concentration in order to activate these CDKs so they can trigger different phases of mitosis. So this is very important. The cell cycle has 3 specific checkpoints where the cell cycle stops by default. So these checkpoints can be overcome only if the proper go signals are received. And what these go signals and these stop signals are basically doing is either activating or inhibiting enzymes of cell division, or they're trying to increase cyclin production or destroy cyclins. So this might sound complicated. There's go signals and stop signals and all these signals. But what these signals are doing is controlling the amount of cyclins inside of the cell. And they're controlling which enzymes are active and which are inactive throughout the cell cycle. So there's a lot of cell signaling going on. So pause here and think about what's the advantage of a checkpoint? What's the point? Why stop the cell cycle? Right? Wouldn't it be more efficient if I didn't have a checkpoint so I could speed up cell division? Like, why would I want to build in a stopping point? All right, so the answer is, just like in an airport security checkpoint, you want to make sure nothing is getting where it shouldn't be, right? We want to make sure nobody dangerous is going to go on the plane. So you need security checkpoints at every step of the way to make sure that we're screening, to make sure everything is okay before we move on to the next step. And that's what the cell does. It's going to make sure everything is okay before moving on to the next step. So we said the cell cycle is under tight control. The checkpoints are what keep it under tight control. So specifically, there are 3 checkpoints. So at the G1 checkpoint, that's the first checkpoint, the one in between G1 and S. So after G1, there's this G1 checkpoint. And this is the checkpoint that says, are you either going to go on to S phase, or are you going to go into a G0 phase, which is non-dividing. G0 or G0 is non-dividing. Those cells are like our neurons. They are senescent, meaning they don't do mitosis anymore. So they never get, certain senescent cells never get past this checkpoint. But at this G1 checkpoint, we're basically saying, is our DNA damaged? Right? And why is that important at this phase? Why do we care if DNA is damaged here? Well, we're about to make copies of it in S phase. So if there's mistakes, right, if there are typos in DNA over here, or if there's any damage in the DNA, why would I want to make a copy of it? Right, so that's that first checkpoint. Is DNA damaged? After DNA is replicated, there's the S phase checkpoint, which is this is like a fake checkpoint. It's not really considered one of the 3, but we're making sure that DNA is replicated correctly. So this one you don't have to know as much. These are the 3 you have to know. So there's the G1S. The G2M checkpoint is very important. This is right before mitosis. So this is saying, has all the DNA replicated properly? Did all of the DNA damage get repaired? And is all of the spindle-making machinery, the microtubules, put in place? This is the last stop before we enter mitosis. Within mitosis, there is a very special checkpoint called the metaphase anaphase checkpoint. What do you think is being checked here? Right? We want to make sure that every chromatid is attached to a microtubule. Right? We want to make sure that all the chromosomes are properly aligned in the middle of the cell. Because we don't want to pull chromosomes apart unless they're all lined up equally and in the right position. Right? So this is very, very, very important, this metaphase anaphase checkpoint. If they're not properly lined up, or there aren't microtubules attached to every chromatid, you're going to have a cell division that results in unequal chromosome numbers. You might have one with 46 and you might have one with 23. So checkpoint arrest is really important, right? We can stop the cell cycle if necessary. Right? In G1, that checkpoint made sure, is there any damaged DNA? Is the environment just right? If not, we're going to stop the cell cycle in G1. If there's any incompletely replicated DNA or damaged DNA, that again will be stopped. If the chromosomes are improperly attached to the mitotic spindle, again, that will be stopped. Whenever you see these arrows, that means inhibiting. So that might be new, but if you see a line like that, that means inhibits, as opposed to an arrow like that means activates. That's just good to know. So here's a review of the 3 checkpoints that are brought up in your book. Right, the G1 checkpoint makes sure that DNA isn't damaged, so it's committed to duplicating. The mitosis checkpoint makes sure that all of the DNA has been replicated properly. And then within mitosis, you have the metaphase anaphase checkpoint, which is called the spindle assembly checkpoint. You want to make sure that all the chromosomes are attached to the microtubules and that they're properly aligned in the center of the cell. So let's watch a quick video on cell cycle checkpoints.
Cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
And just as a note, there is no C phase mentioned in your book. So this animation mentions C phase for cytokinesis, but the book includes cytokinesis in M phase along with mitosis. So now that we spoke about cell growth and division, we have to talk about this necessary balance that there must be for all cells to maintain homeostasis. Right, there must specifically be a balance between cell division, which includes mitosis and cytokinesis, and cell death. Right, so what can happen, right, if we tip the scale differently, right? We'll talk about how disease can result if we tip the scale either way. We want to make sure that in a healthy individual, as cells die, they are replaced, right? And vice versa, right? We don't want to have too many cells dividing and surviving. We need to have a balance between cell death and cell division and cell survival. So now we will talk about apoptosis, which is programmed cell death or cell suicide. So you pronounce this word apoptosis. The PT is silent, like pterodactyl. So apoptosis is a very good thing, actually. So this is the one case where suicide is not terrible, right? This is programmed cell death. They are controlling when they kill themselves, and it's for the benefit of the rest of the organism. So apoptosis is a very tightly controlled process. It's called programmed cell death because it's programmed. There's very, very fine-tuned steps that take place to kill the cell. This is different. There's another type of cell death called necrosis, where the cell kind of just explodes and it's very messy. Not a good cell death. Apoptosis is programmed, controlled cell suicide. As embryos develop, they need to constantly do apoptosis. So when this tadpole becomes a frog, all of the cells that make up the tail have to kill themselves at a certain point in metamorphosis. So all of these tail cells are programmed to kill themselves so the tadpole can become an adult frog. In neurons, so in our brains, for example, neurons try to attach to each other, but neurons that do not have the proper attachments automatically kill themselves. So this is called neuronal pruning, right? So if a neuron is not attached to a proper destination, it will kill itself. Believe it or not, we all have webbed feet as we develop. But what happens is the skin cells in between our digits undergoes apoptosis, right? So we kill all the cells in the webbing, and that's how we have separate fingers. You can see over here, this is looks like polydactyly, which is when digits are fused together. Right? Apoptosis did not occur here like it should have. Right? Apoptosis should have occurred here to kill these cells. So that's as we develop. But right now, some of your cells are undergoing apoptosis too. Our bone marrow and our intestinal cells constantly need to be renewed. So our bone marrow, our blood cells always have to be replenished. So old cells are dying by apoptosis all the time. right? We want to kill cells so we can make new, fresh cells. Same thing with our intestinal cells. After a certain period of time, they're told to kill themselves, right? They'll stop dividing. They will never enter mitosis again because we want fresh cells to start being developed. And most importantly for this lecture, we'll talk about apoptosis being really important and killing a cell that has a genetic mutation. So any genetically mutated cell could be very, very dangerous for the organism. So those cells are told to kill themselves right away. So apoptosis is triggered by enzymes called caspases. So here's normal cells. When a cell is ready to kill itself, it rounds up and the nucleus starts to fragment itself into different pieces. And then what's called blebbing occurs. So these little blebs are little bubbles, like almost vesicles of plasma membrane. So all of these cells are fragmented until there's nothing left. So it's very tightly controlled. So caspases are enzymes that can chop up larger molecules into fragments. So now we have all these different cell fragments have pieces of the nucleus, but they're not functional cells. So we kind of chopped up the cell in a food processor. And again, mitosis and apoptosis are opposing forces, right? Mitosis will increase cell numbers, apoptosis will decrease cell numbers. So all of this is required for homeostasis. Here's a quick animation on apoptosis.
Under the microscope, we can see how cells are destroyed. This process continues throughout our lives as cells become damaged or just worn out.
So again, you could look at the blebs actually, right? Those are the fragments.
Or just worn out.
So here's a cell and there are its fragments. So very, very tightly controlled process that happens all at once. So I want to take a step back. So now that we spoke about cell growth and mitosis, we spoke about cell death. Let's go back to the very, very beginning of the cell cycle, which is the G1 checkpoint. So a functional G1 checkpoint is critical to maintain the balance between cell life and cell death. Because at the G1 checkpoint, the cell has to make the critical decision. Am I supposed to divide? Or am I supposed to wait? Or am I supposed to kill myself? That's what a cell is asking itself at the G1 phase. So the only way that it will go on to G1 is if a go signal is received. And if a cell receives a go signal at the G1 checkpoint, the cell will continue on to which phase? S phase, right? That's where the DNA will be copied. However, if a cell does not receive a go signal at the G1 checkpoint, the cell will go into a non-dividing state. It will be on pause. It will be put on pause. Or it may kill itself in apoptosis, if it's genetically mutated or if it's no longer needed. So this G1 checkpoint is the starting point, right, for whether you're going to die or live. So this checkpoint is very, very, very important. And this checkpoint we said is critical for maintaining balance, right? The cells have to be told, are they supposed to die or are they supposed to live and divide? So what happens when things become imbalanced, right? What happens when you have too much cell death and not enough cell division, cell growth, and cell survival? Well, you can have diseases like Alzheimer's or Parkinson's, right? Those are neurodegenerative diseases, meaning the neurons are not developing properly enough, they're not connecting properly, and they start dying. So there's too much cell death in neurodegenerative diseases like Alzheimer's and Parkinson's, right? Not enough cell growth and survival. And of course, you could have the other case where you have way too much cell growth, division, and survival, and not enough cell death. And that results in cancer. So this one slide shows us why it's so important to understand the cell cycle. right? If we understand the cell cycle and we can understand how a cell controls whether it divides or dies, we could potentially come up with better treatments for Alzheimer's and cancer, right? Both of these are diseases of improper cell cycle. That's all it is to it, right? So if we can figure out more information about the cell cycle and we can understand how these checkpoints work, maybe we can design a cure for these diseases, right? To help get the cell cycle back in balance. So for the rest of this talk, we'll discuss cancer. So we said that cancer is what results when things become imbalanced and there's way too much cell division, growth, and survival, and not enough cell death. So what is cancer? Cancer is characterized by uncontrolled cell growth and the spread of abnormal cells, resulting in cell masses called tumors. The key word here is uncontrolled, right? There's uncontrolled cell growth. So over here, you can see that there are clear differences between these tumor cells that stain differently and normal cells. These tumor cells have a lot of genomic instability. They change their size and shape. You can see over here, there are these growths. These are called tumors, right? And these are quite large. These are very large tumors in this tissue. So cancer cells look and act very differently from healthy cells. And cancer is not a genetic disease, quote unquote. So a lot of people say, oh yeah, cancer is genetic. But in fact, cancer is more of an age-related disease than a hereditary one. Yes, there are cases of inherited cancers, right, like in childhood cancers. You'll read a lot more about that, hopefully, in the relevancy module on cancer. And you can have a predisposition to developing cancer. So certain risk factors can run in families. However, for the most part, cancer is a disease of old age and not genes. So you can see in this graph, as age increases, so does the incidence of cancer, almost in a very linear fashion. Especially you can see prostate cancer is one of those that really gets more prevalent in older men. So what causes cancer? What we know so far is that cancer is uncontrolled cell growth. So what can cause that? There's a very simple answer. Genetic mutations. Genetic mutations are what lead to cancer. And a genetic mutation is an alteration in the genome's genetic instructions. So it's basically like the instruction, right? Imagine the DNA is like an instruction booklet. Tells your cells what to do and how to do it. But if there's a typo in the instruction booklet, right, it might be really hard to follow those instructions. So a mutation is like a DNA typo. What can cause a typo, right? What can cause DNA to be mutated? Chemicals called carcinogens or mutagens can alter genomic DNA. So a mutagen is anything that can mutate DNA. A carcinogen is a mutagen that causes cancer. So carcinogen comes from the word for cancer, which is karkinos, which comes from the Greek, I believe, for crab, because some of the tumors looked like little crabs to the person who named them. So carcinogens include cigarette smoke, right? So far we said cancer is a disease of uncontrolled cell growth that's caused by genetic mutations, and carcinogens, chemicals, can mutate your DNA. So cigarettes are very carcinogenic. There are 250 known harmful chemicals in tobacco smoke. Of those 250 harmful chemicals, at least 69 can cause cancer. And this graph shows the per capita cigarette consumption. So this shows that how the prevalence of cigarettes increased and the result on lung cancer in males and in females. So you can see directly following the rise in cigarette consumption, you see an increase in lung cancer. So it's a very high correlation. You can see that, interestingly, there's a lag in females. So females did not dip. So you can see over here that in the 90s, there was a decrease in male lung cancer. So for females, they were overall, in general, slower to start and slower to finish. So slower to quit smoking. So right now, there are more female smokers. There's a lot more female lung cancer more prevalent than any time in the past. So lung cancer rates lag the smoking rates by about 30 years, though. So where the height, of cigarette smoking was in the 60s, the height of lung cancer was in the 90s. So it takes some time for mutations to really take effect on your health. So we said that carcinogens can mutate DNA, but what else? So UV light and radiation can actually break DNA strands. So UV light from sun tanning, especially in a sun tanning bed, that UV light can break your DNA. Radiation, we said, is a form of radioactivity that can be used to directly damage DNA. Radiation naturally comes from the sun, just like UV light, but we're protected by ozone and some CO2 in the environment. So we're not directly bombarded by these UV light, but upon extended exposure, if you're sitting outside in the sun all day long, eventually UV light will mutate some of your genes. Also, viruses can insert their DNA into your cells, and you can imagine, let's say the virus genes gets inserted in the middle of one of your genes. That's going to cause a major typo, Imagine copying and pasting a random genome into your instructions manual, The instructions cannot be followed if there's a random sequence inserted in there. So let's go from the beginning. We said cancer is a disease of uncontrolled cell division that's caused by genetic mutations. Genes can be mutated by carcinogens like chemicals, cigarette smoke, but they can also be damaged by viruses or by UV light or radiation. But the next question becomes, what kinds of genes are mutated in cancer? Is it just any old gene that gets mutated and develops cancer? It actually turns out not to be the case. So there are specific genes that get mutated in cancer. If you remember, we said the cell cycle is regulated by a control system. We said that there are specific checkpoints where the cell cycle arrests until a go signal is received. There are two types of genes that control these cell signals for proper cell division. This is very important. Proto-oncogenes. Proto-oncogenes are genes that promote cell growth and division. So a proto-oncogene is a gene that encodes for a protein that tells the cell to divide. Those are the go signals, Like the growth factors. So growth factors normally signal to the cell to start dividing. That's called a proto-oncogene. Those are healthy cells that tell the cell when to divide. Those are healthy genes that tell the cell when to divide. The other type of gene that's critical for proper cell division are called tumor suppressor genes. And tumor suppressor genes are genes that inhibit cell growth and division. So a tumor suppressor gene is something that will stop the cell from entering mitosis and might tell the cell to actually kill itself. So apoptotic factors, things that tell the cell to die, are controlled by tumor suppressor genes. So what we're going to see is that if there's a typo in a proto-oncogene or a tumor suppressor gene, then we're in trouble. Right? It's these two types of genes, these go signals and stop signals, that cannot be messed around with. Because if you mess around with those, the cell cycle will not be regulated and it will be uncontrolled. Right? And we said that's what cancer is. Cancer is uncontrolled cell division. So we said proto-oncogenes are the genes that promote the cell cycle. They say go, divide. However, when a proto-oncogene is mutated, so when we change the genetic sequence and the instructions are not good anymore, we call that gene an oncogene. So oncology is the study of cancer. So an oncogene is a cancer-causing gene. A proto-oncogene is a healthy gene that tells the cell to go and divide. When you mutate a proto-oncogene, we then call it an oncogene. And an oncogene leads to uncontrolled cell growth and division. An oncogene will continually tell the cell to divide, even when it should not be dividing. So here we start off with a nice, healthy proto-oncogene that tells our cell to go and divide when it needs to. However, some kind of cancer-promoting agent mutated the proto-oncogene, so UV light or chemicals, something, changed the proto-oncogene into an oncogene. And that oncogene told the cell to divide and grow, even when it shouldn't. And that's what a cancerous cell is. The other type of genes that control the cell cycle are tumor suppressor genes. And tumor suppressor genes are the checkpoint genes that prevent cell division unless it's required. So when tumor suppressor genes are mutated, there are no more stop signals to tell the cells to stop dividing. Right? We said that there are checkpoint genes that tell the cell to stop dividing unless another signal is sent. Well, let me show you this example. So normally there's a tumor suppressor gene that encodes for a protein that stops cell division. So this is a growth inhibiting protein that can stop cell division, keeping it under control. If I mutate that tumor suppressor gene, I'm not going to be able to make a functioning inhibiting protein. Right? There's no stop sign I can make. So cell division will not be under control and it will continue to divide. Right? So when a tumor suppressor gene is mutated, there's no more stop signals to tell the cells to stop dividing. One tumor suppressor we should all be very thankful for is called P53. One of the most important genes to understand. P53 is called the guardian of the genome. And it's a very important title because it has a very important role. P53 is activated by DNA damage. This is a really cool aspect automatically. So it is activated by DNA damage. And when P53 is active, it can stop the cell cycle and initiate DNA repair. So this is really, really cool, right? So this is anytime there's a mistake in DNA, there's an enzyme called P53 that gets activated and will tell the cell cycle to stop. That's a stop sign. Remember, the tumor suppressor genes are like stop signs. So P53 is a stop sign that only gets activated by DNA damage. So it's like, I'm trying to think of a good example. So many different examples you could come up with, but it's almost like quality control. So there's like a scanner that looks for any defects. And when there's a defect, like let's say you're making some kind of a product and it's going down the assembly line and there's this guardian, right? And if there's any type of defect in any of the products being made, the whole assembly line will stop and something will be activated that will either kick that product off the assembly line or alert the manufacturer to fix it, right? It's like a built-in system. I don't know if that made so much sense, but it's a built-in system that gets triggered by DNA damage and then will prevent the cell from dividing with that DNA damage, right? So if the DNA cannot be repaired, P53 can initiate apoptosis. So, in response to DNA damage, P53 can cause cell cycle arrest. It will try to get the DNA to be repaired, or if not, it will cause apoptosis. You can see if you have a mutant P53, that won't be good, right? So think about what would happen if you don't have P53. So don't even look at the rest of these words, but what would happen if you don't have an active P53? Right? Even if there's DNA damage, the cell cycle will not arrest and the DNA will not be repaired. You're just going to be spreading damaged DNA. Right? So this is a very important gene, and it's no surprise that in cancer, P53 is commonly lost. More than 50% of all tumors of all types have mutated P53 proteins. This is why P53 is called the guardian of the genome, because without it, there's nothing to guard our genes, and they're just free to be mutated. So we're very thankful for the tumor suppressor P53. So here's an analogy that might help. So when you drive a car, all cars undergo everyday wear and tear, right? You might bump into something, you might get a little scratched, but most of the time it's not going to impact the car itself, right? And as the car gets older, it's just going to accumulate more scratches and dents, but most likely it won't be fatal, right? It won't cause the car to kill you. But what can mechanically lead to a fatal car accident? If the brakes don't work, right, that means you could drive right off a cliff without brakes. If the ignition is always on, same idea, you can drive right off the cliff if the ignition is always on. So let's try to bring this back to cancer. So a cancer cell, not a cancer cell, your normal cells undergo everyday wear and tear, right? We sit out in the sun, we might be exposed to some chemicals. So every day our genes are being mutated. But most of the genes that are being mutated aren't involved in cell division, right? It's only when the brakes, which are tumor suppressor genes, or the ignition, which are proto-oncogenes, are mutated, that the car will be killing you, right? The car will definitely cause you to be in a car accident, right? If the brakes don't work, right, meaning the tumor suppressor genes don't work, the cell will just keep on dividing with nothing to stop it, right? Normally, we want to have the ignition only on when we need it. So proto-oncogenes are good. But if the ignition is always on, that's like an oncogene, right? It's always telling the cell to divide. So again, most of our DNA gets mutated and will either be fixed by our immune system, or taken care of by the immune system, or it won't be in an important gene, so the cell can live with it. But if we have a mutation in tumor suppressor genes or proto-oncogenes, then we can lead to uncontrolled cell growth, which is what cancer is all about. So another tumor suppressor gene I'd like you to be familiar with is BRCA1. There's BRCA1 and BRCA2. And gene mutations in tumor suppressor genes like BRCA1 increase the risk of breast cancer. So I said before, cancer is not a genetic disease, right? It's not like you have this gene, you get cancer. But there are some tumor suppressor mutations. So there are some versions of BRCA1 that are mutated that do not function like proper tumor suppressors. So therefore, those individuals are more likely to develop cancer, right? Because there's no stop signs, right? There's no brakes. So BRCA1 is inherited, which makes it more prevalent in certain ethnic groups. Specifically, very high prevalence in Ashkenazi Jewish populations, and way more so in Hispanic populations than others. So this is an example of how cancer risk can vary by ethnicity, and how different ethnic groups are more at risk for different types of cancer. So reducing DNA damage reduces cancer risk, right? So mutations are caused in many ways. We said it can be inherited, so that's one thing you can't really control. But there's a lot of factors in the environment that damage DNA and that increase cancer risk. So we have a lot of control over at least reducing the risk of developing cancer in our lifetime, right? By eating a healthy diet, right? Avoiding obesity, getting very good exercise, do not use tobacco or sun tanning beds. Always practice safe sex, and make sure that you do as many diagnostic tests, right? Get blood work done regularly, go to the dermatologist to get screened for any skin cancer. So it's really about early detection, right? Making sure you detect it as early as possible so you can treat the cancer. Which of the following is not a cancer-causing gene? So the answer is D, right? A and C are not cancer-causing. Only an oncogene is cancer-causing. Which of the following may damage DNA? You could pause here. The answer is D. All of the above may damage DNA. So far we said that cancer is uncontrolled cell growth. But why is that so bad? Right? So what is actually deadly about cancer? And why do we care if we just have extra cells? Well, it's metastasis that makes cancer so deadly. So cancer cells metastasize, meaning they spread through tissue, through our bloodstream, and they can then form tumors in other parts of the body. This is why cancer is so deadly. So what can start as a tumor in situ, which means a tumor in place, it's localized, it can invade surrounding tissues and enter our blood vessels and our lymph vessels. Once it's in our circulatory system, our blood and lymph can transport these little cancer cells to other distant sites in our body, and the cancer cells can reinvade and grow at these new locations. So they kind of colonize the body, traveling through the blood vessels, the lymph vessels, to try to find new places to form new tumors. So in this way, one little tumor can spread throughout the entire body and disrupt a lot of body functioning. So why is skin cancer so deadly, for example? Right, what starts as a little mole, or a little growth on your arm, can metastasize. So it's a melanoma because it started on your skin, right, in a melanocyte, which is one of the pigment-forming cells of the skin. But it can metastasize, the tumor can metastasize to the liver and the brain. And once it's in the liver and the brain, then it's deadly. So because of metastasis, cancer is very deadly. And you might not think, oh, skin cancer, who cares about skin cancer? It sounds so, benign, which is a word coming up next. But skin cancer is very deadly because that skin cancer can spread to your brain. But it's still referred to as a melanoma. Even though it's in your brain, you don't call it brain cancer. It's still called a melanoma because that's where it started. So there are two types of tumors, because not all tumors are cancerous. So if you have a growth that does not spread, we call that benign. A benign tumor is not metastatic. It cannot spread. Right? Like Ben, B-E-N, means good, right? Like benevolent. So benign tumors are usually encapsulated, so they do not invade neighboring tissues or spread. Malignant tumors, mal means bad, like maleficent, right? Malignant tumors invade tissues, enter blood cells, and metastasize. So malignant tumors are the ones that are very deadly. And over time, a benign tumor can become malignant. So it starts off as one mutated cell, can grow and accumulate, grow and accumulate, grow and accumulate over time, and then it can enter the bloodstream and start metastasizing. So one cell acquires a mutation. So here we have one brown cell over here has the ability to start a tumor. But this one mutation isn't enough to cause cancer. Tumors accumulate multiple mutations as they spread. And this is called the multiple hit hypothesis. It's not enough to just have one mutation and get cancer. It's actually an accumulation of multiple mutations in different genes that are responsible for cell division that causes carcinogenesis. So here we had one cell that has a mutation, so this keeps on growing and growing into a colony of cells. So here we have a group of brown cells. One of those brown cells mutates even further, has a separate mutation, right? So this is now heterogeneous, meaning this purple cell, cancer cell, has a different set of mutations than the rest. Now these purple cells will keep on growing, and now they can invade lymph vessels or blood vessels, right? Because that mutation gave them an advantage to divide and invade even more. So they keep on acquiring new mutations, more advantages to invade. And over time, right, they keep on being able to make more mutations, delete DNA, add DNA. They're out of control. Cancer cells are completely out of control. So that allows them to spread to other locations in the body. Tumor cells completely alter their metabolism. So they do cell respiration in a different way. They duplicate their DNA with a lot less fidelity. So tumor cells are very different than healthy cells, but it's an accumulation of multiple mutations that makes cancer progress. Something that tumors can do that makes them really deadly is called angiogenesis, which is the formation of blood vessels. So tumors can actually form their own blood vessels, which can help nourish the tumor and help them increase in size and spread farther. So tumors secrete a chemical called VEGF, vascular endothelial growth factor. So it's a growth factor that the tumor secretes, and those tell our blood vessels to grow into it. So this is a huge adaptation for tumors. So tumors can basically tell our own blood vessels to come closer. So then our tumor will have an increased blood supply. This is what helps tumors grow and helps tumors metastasize, right? By releasing this chemical signal, our blood vessels will grow right into the tumor, just like the tumor wants. Without angiogenesis, cancer cells will die due to lack of nutrients. So you can take a second to look at this case study. Endostatin is a drug that inhibits angiogenesis. And on top, mice were treated, and on the bottom, humans were treated. So what you're looking at is the tumor volume, and endostatin was administered on the days in the brackets. So where this is a little break, right? These are the treatment days, are the ones in brackets. So does it work in mice? Does it work in humans? What do you think? Pause here. So these results are pretty interesting. You can see that as the mice were treated with endostatin, there was a huge reduction in tumor volume, almost eliminated completely. But when treatment was stopped, the tumor grew back even bigger in mice. When treatment was resumed, it went back down again. So up, down, but after 4 treatments, they saw a huge decrease in tumor volume in mice. All right, grew a little bit again, but after 4 treatments, then you saw a huge decrease in tumor volume. So it seemed to work in mice. In humans, it wasn't as good. There was definitely a difference compared to the control. This is the negative control, untreated. There's just a great increase in tumor volume. What endostatin did in humans was delay the growth of the tumor, but in the end, the tumor ended up being the same size. There are other chemotherapies that work very well to inhibit angiogenesis. There's a drug called Avastin that blocks activation of the VEGF receptors, so they cannot do angiogenesis. So if this is a cancer cell, this is a blood vessel. VEGF is normally on the blood vessel and will tell the blood vessel to grow. But if this is blocked, right, if the VEGF receptor is blocked, that means the signal from the cancer cell can never be transmitted into the blood vessel, right? So Avastin inhibits VEGF, so angiogenesis cannot occur. So Avastin is a very successful chemotherapy for certain tumors because it blocks angiogenesis, so the tumors can't build their own blood vessels. So, as we've said, all this time, cancer cells are very different from healthy cells. Something else that cancer cells lose is contact inhibition. Normal cells stop growing when they contact each other. Cancer cells keep on growing on top of each other in clumps. Telomerase is an enzyme that's overexpressed in cancer cells. And telomeres, which are the ends of chromosomes, gradually shorten as healthy cells divide. And what happens is when a cell has too short of a telomere, it just stops dividing and it dies. However, cancer cells don't do this. They don't die when their telomeres get shortened because they have an enzyme called telomerase that constantly lengthens their telomeres. So telomerase always extends the telomeres in cancer cells, making cancer cells essentially immortal. They keep on dividing. Right? If their telomeres never shorten, they never die. So telomerase is overactive in cancer cells, just like it is in stem cells, which makes them always able to divide. So taken together, there are 6 hallmarks of cancer, and these are very important to understand. And I have a summary slide on the next 2 pages, or 2 summary slides, I should say. But overall, cancer cells resist cell death, right? They resist apoptosis. They sustain proliferative signaling, meaning they always are told to go and divide. They evade growth suppressors, right? They don't listen when they're told to stop growing. They induce angiogenesis, right? They form blood vessels. They activate invasion and metastasis. And they are immortal, right, because of their telomeres. So these are the 6 hallmarks of cancer cells. So I'm not going to repeat all these things. It's a little wordy, these slides. But cancer cells are non-specialized, meaning they don't look different from another. They keep on dividing and dividing. They have really messed up genes and chromosomes. They don't kill themselves when they should. So normally the immune system will recognize a cancer cell and tell it to kill itself. But cancer cells are sometimes just so abnormal that they trick the immune system. And the immune system can't kill these cancer cells. Right? We said that they form tumors. They undergo metastasis and angiogenesis. So pause here. And which of the following is not a hallmark of cancer cells? So what is not a hallmark of cancer cells is C. Right? Cancer cells do not have reduced rates of mitosis. Right? They have increased rates of mitosis. So here is a summary slide from the book that talks about the causes of cancer. So we said that mutated genes cause cancer. Those can be caused by radiation or viruses, hereditary causes, or other carcinogens like pesticides or something, right? We also said that a growth factor is a protein that activates signaling pathways that can extend to the nucleus. And a proto-oncogene, right? A proto-oncogene is a gene that codes for a growth factor, a receptor protein, or some kind of a molecule that's involved in cell signaling to tell the cell to grow. So proto-oncogenes are gene products that promote the cell cycle. A tumor suppressor gene does the opposite, right? A tumor suppressor gene codes for a signaling protein in an inhibitory pathway. So if a tumor suppressor gene is mutated, then we cannot tell the cell cycle to stop. Okay? So that's why we need both the combined action of healthy proto-oncogenes and tumor suppressor genes to make sure that the cell cycle is under tight control.
Cancer is a group of over 100 diseases characterized by abnormal, uncontrolled cell growth. In a healthy body, cells grow, die, and are replaced in a very controlled way. Damage or change in the genetic material of cells by environmental or internal factors sometimes results in cells that do not die and continue to multiply until a mass of cancer cells or a tumor develops. Most cancer-related deaths are due to metastasis, malignant cells that penetrate into the circulatory system and establish colonies in other parts of the body. Great advancements have been made, but cancer is still the leading cause of death for people under the age of 85. In the United States, 1 in every 4 people die from cancer.
So now I want to talk about some extra information that won't be covered. So anything you see here won't be on the exam or the quiz, but it's good to know. So cancer is named according to the type of tissue in which it first forms. So sarcomas are cancers of bone and cartilage. Carcinomas are cancers that arise from the epithelium, which are it's tissue that's one cell thick, so like skin or the lining of organs are all epithelial lining. And if some of those epithelial cells grow out of control, that's called a carcinoma. A lymphoma is cancer of lymph tissue or immune cells. Leukemia is a cancer of stem cells, those that produce white blood cells. So when a person is diagnosed with cancer, a variety of weapons are available to combat it. So we're familiar with chemotherapy and radiation. Those both kill cancer cells. Radiation uses ionizing energy to specifically target and kill a tumor by breaking its DNA. So remember that radiation can damage our DNA, but we can use that to our advantage in a way. We can make very, very, very high levels of radiation and target them at a tumor cell, so we can selectively kill tumor cells. Surgery is also very common, where you can just physically remove the tumor and all the surrounding tissue, so it can't metastasize anymore. Chemotherapy drugs are used to kill cancer by disrupting some aspect of cell division. So chemotherapy usually works by preventing the cells from dividing anymore. What are some side effects of chemotherapy? So when you think about chemotherapy, what are some of the known side effects? So hair loss is very common. Also, nausea is very common. So what does hair loss and nausea have in common? What's the commonality between the two? It's a little tricky, but hair cells are cells that divide very quickly. They're always growing very fast. Similarly, your intestinal cells and your gut cells are always dividing very fast, right? We said they're always undergoing renewal. So chemotherapy targets any cells that divide quickly. So your hair cells divide very quickly, those are going to fall out. Those are not going to be able to divide anymore because of the chemotherapy. You're going to have nausea because your gut cells are constantly killing themselves. They're being killed. Right? So there are side effects to chemotherapy because it targets healthy cells too. Right? And the cells that divide most frequently are most affected by chemotherapy. So what cellular structure can be targeted to specifically disrupt cell division? Right? Like what part of the cell would I have to stop in order to prevent the cell from dividing? So think about that. So hopefully you thought of microtubules, right? Microtubules form the mitotic spindle that pulls chromosomes apart. If chromosomes can't be separated by the microtubules, then cells can't divide. So we can use a drug called Taxol. It's actually a chemical that was found in the bark of a tree, of the yew tree. So in this bark of a tree, they extracted a chemical called Taxol. And Taxol, which is shown here in this little red part, this little red molecule, it binds to microtubules, preventing them from growing or shrinking. So remember that microtubules are made of tubulin. Scientists discovered the 3D shape of tubulin and figured out that Taxol, this chemical, can bind to tubulin and prevent the microtubules from growing or shrinking. If microtubules can't grow or shrink, that means they can't grab onto the chromosomes and pull, right? So this is a really fundamental, this was one of the first chemotherapies to come about because it can prevent cells from dividing. So at which checkpoint does Taxol arrest the cell cycle? The answer is, right, D, the metaphase-anaphase transition is where Taxol will arrest the cell cycle, right? Because in anaphase is when those microtubules shorten and pull on the chromosomes. So they're going to be stopped, they're going to be halted at that metaphase-anaphase checkpoint. As we go into the future, there's more and more hope to treat cancer. So as we see more genetic technologies become cheaper, we're going to be able to screen the entire genomes of patients and figure out the exact mutations. So new technologies can screen the entire genome of a patient and determine the exact mutations that can lead to cancer. And you can help pinpoint. You can say, oh, if you have mutation X, we know that chemotherapy Y is best for you. Or if you have this mutation, then we know that statistically it's better to do this type of treatment. So as we get more and more data, we'll be able to do what's called personalized medicine, right? You can look at somebody's genes and personally tailor their chemotherapies based on the mutations that the patient has. There's also new therapies that can take a cancer patient's immune cells out of their body, and you can reprogram them so they can target cancer cells. This is called CAR T. This is a new type of therapy. So you can take a cancer patient's cells and reprogram them outside of the body, and you put them back in the body so they can kill cancer cells. This is very cool stuff. There's another type of therapy that's coming out that uses viruses that kill only cells that don't have P53. Remember, if a cell doesn't have P53, it can keep on dividing uncontrolled. So we could use a special virus that kills cancer cells only that do not have P53. And there are 2 links down here that will help walk you through some of these treatments. So it's very cool stuff. Here is a final animation I'd like you to watch.
Every year, it is estimated that there are over 12 million new cases of cancer worldwide. The type of therapy that a doctor will recommend depends on the type of cancer, how far it has progressed, and where in the body it is located. But the majority of cancer treatments fall into one of 3 categories: chemotherapy, radiation, and surgery. Chemotherapy kills or slows the growth of cancer cells within the patient's body using drugs. It is a more likely choice when cancer is located in several parts of the body. Radiation also kills or damages cancerous cells. Particular types of focused energy, called ionizing radiation, are directed at the site of the tumor. The DNA of the cancer cells is damaged, preventing them from replicating. This may slow the growth of the tumor, stop it, shrink the tumor, or kill cancer cells that remain after surgery. Various methods are used to direct the radiation at only the cancer cells, minimizing damage to nearby healthy tissue. In the case of surgery, the intent is to remove the tumor rather than to kill the cancerous cells that make it up. The potential benefit and risks of surgery will depend greatly on the type of tumor, its location, and a number of other factors. Because it involves cutting into the body, surgery almost always entails some side effects, although some newer minimally invasive techniques, such as laparoscopic and robotic surgery, can help to reduce them.
So I want to end off by talking about some cancer statistics. You can click on this link for the most up-to-date stats, but I want to show you, these are from a few years ago, but it's pretty much the same. So leading new cases in males are prostate cancer, right? So 20% of all new cases of cancer in males are prostate. In females, 30% of cancer is breast cancer. In both males and females, the most deadly cancer is lung cancer, followed by prostate cancer in men and breast cancer in females. So this is a really interesting chart that looks at one axis of estimated new cases. So these are the most common, these are the least common. And on the y-axis, you have the least aggressive and the most aggressive. So the farther up, that means the most deadly. And the farther right, that means the most common. So over here, we have high mortality, very common cancers like lung cancer, right? Those kill a lot of people because they're very common and they're very aggressive. Over here, you have some very, very deadly cancers, but they're very rare. So like esophageal cancer and pancreatic cancer are much more rare. So not as many people have it, but they're very deadly. So different pharmaceutical companies might want to go for trying to cure a high mortality common cancer like lung cancer. I think almost every project I was on when I was working in pharmaceuticals was related to lung cancer. It's very common. Other companies might try to design a drug for pancreatic cancer, which is very rare, but it can make a lot of money and save a lot of people's lives. Or not a lot of people, but save the few lives with this very rare cancer. So the good news is cancer death rates are decreasing overall. So this is overall good news in the fight against cancer. So pulling it all together, we said that if mitotic cell division is uncontrolled, it leads to cancer. All right? Mitotic cell division has 3 stages: interphase, mitosis, and cytokinesis. Replication occurs in interphase, right? Mitosis is division of chromosomes, which occurs in the following phases, and cytokinesis is the division of the cytoplasm. So that is the end of the lecture. If you're interested in learning more about cancer, I would recommend this book, The Emperor of All Maladies by Siddhartha Mukherjee, and it's a very, very good book that talks about the biography of cancer. So the whole story of cancer going from thousands of years ago. Another very, very, very good book to read is The Immortal Life of Henrietta Lacks. And I would like for you to watch this this TED Talk. You should click on this link over here. And it tells the story of a woman named Henrietta Lacks. And doctors took her cells without asking. And today, we still use her cells in labs around the world. So again, Henrietta Lacks was a poor African-American woman who had cervical cancer. Long story short, the cells were taken from her, and the cells just kept on growing and growing and growing like magic. We know it's not magic, we know it's cancer, and because of the properties of cancer cells allows them to grow and grow and grow. But these cells were remarkable, and we still use these cells today. They're still alive. And we grow them, and they were shipped around the world to be tested on. These cells were even sent into outer space to test the effects of gravity on human cells. So this is how Henrietta Lacks is immortal in a way, because we're still using her living cells today. I've used them many times. Every biologist has likely used HeLa cells. They're called HeLa after Henrietta Lacks. So now we'll end off with this video, this picture again. The answer was I stained the nuclei, the DNA, in blue, and in green are the microtubules. So here you can see, well, you should tell me, what phase does this look like? Right, this is like anaphase or telophase. Right, here you can see the 2 nuclei are separated. So this looks like anaphase. And if you're interested, these little red dots are the centrosomes. So you can see these 2 dots represent the centrosomes that are pulling on the microtubules, so the DNA can be separated to opposite ends of the cell. So that is the end of Chapter 9. Next time we'll talk about meiosis, which is how sex cells like sperm and egg get made.